see me and hear me well. But yes, my name is Alina, and I am a second year PhD student at the University of Oxford, and specifically at Balliol College. And my background is actually in biochemistry. So I did my bachelor's in biochemistry at UCL. And then I came to Oxford to do a master's in genomic medicine. And now my PhD program is called clinical medicine, although it's not very clinical. I'm doing mainly basic research in medical sciences, and I study genetics in the context of cancer specifically. But today I thought I would give you a talk that would be an introduction to gene editing, because when I was thinking of what I would like to study at university, and the reason why I chose biochemistry in the first place is because I was fascinated by genetics and all the emerging technologies that we have in the field. And so, yeah, to start with, I would like to ask you to join a quick poll website if you can. So if you, on your browser, if you just type in vevox.app and then enter this ID, you should be able to enter a live polling session. And this is just an opportunity for us to have a very quick recap of very basic genetic things, and also an opportunity for people who are still joining to join the call. But that shouldn't take long, and I know that it's Tuesday afternoon, so the questions won't be that difficult. Which is, let me see. Which base forms a bond with adenine in DNA? Hopefully you guys should be familiar with this. And yeah. Well, I can see that 19 people have voted out of 21. Excellent. Cool. So I'll just stop this question for a second and show results. So yeah, it's thymine, and hopefully you guys know that it's two hydrogen bonds that we have between adenine and thymine in DNA. Then I'll stop this question here and go on to the next one. Which base forms a bond with cytosine in DNA? Oh, guanine. That's right. Everyone got it right. 100% success rate, so that's good. I might also quickly share full screen of the poll so that those who weren't able to join can. Cool. So if we go to the next question, which should be, one second, close this poll. And which base forms a bond with uracil in RNA-DNA duplex? Which hopefully you should all also be familiar with. Cool, seems like most people have voted. So yes, it is adenine. Some of you might remember it can be a bit confusing sometimes because uracil is sort of a replacement of thymine in RNA. So when we have a uracil in the molecule of RNA, it means that it forms hydrogen bonds with thymine. When we have uracil in RNA, it means that when it forms an RNA to DNA duplex, it would form a bond with adenine. While in RNA, we do not have thymine, but instead we have it in DNA. And last quick question, just to know how much you guys know about the numbers and statistics of the human genome. So how many genes are there in the human genome in total that are estimated? So let's see what we can see from the results. Yeah, most people voted for 20,000, which is the correct estimation. But something that needs to be said here is that this number estimates specifically the number of protein coding genes, while we also have genes that encode other molecules, which I can talk about in more detail in a few minutes. But yeah, so that was just a very quick recap for everyone to make sure that we're all on the same page, that the basics of genetics. Yeah. A quick recap of the so-called classical dogma of biology is that we have DNA that gets converted into RNA by the means of transcription, and then to protein by the means of translation. For those of you guys who are interested in going into more biological sciences or biochemistry or even medical sciences more in depth in the future, you might also want to remember that there are also many other things then encoded by DNA, which are not necessarily proteins, which could be molecules, for example, as tRNAs, which are used to bring amino acids to the ribosome during translation, or ribosomal RNA, which is one of the building blocks of ribosome as a machine. And for the purpose of simplicity and kind of applications to humans, I'll mostly be talking about gene editing in the context of genes that encode proteins. But I think, as a general rule, it's also very important to remember that the definition of gene is very broad. And although in the context of A levels, it's usually considered to be a region of DNA that encodes a protein, once you go more in depth in genetics, you also find out that there are many, many other things and non-protein coding RNAs that play very important roles in the cell. So when we talk about gene editing, I think it's also very important to understand how things could go wrong in the genes in the first place. And here there is a very simplistic diagram of the things, of mutations that we can have in DNA. So at the top, you see the normal sequence of DNA, which can then be copied onto an mRNA and then copied into a sequence of amino acids. However, we can sometimes have substitutions in DNA. For instance, here, C was replaced by T, which led to a change in an amino acid. So the protein sequence is still the same except for this one amino acid. And in most cases, such substitutions did not change much. So either the amino acid that is being changed has very similar properties to the original amino acid, or maybe the original codon encoded the same amino acid that was there before, which is not the case here, but this can also happen. So we can have silent mutations in this case as well. Then we can have insertion, which is where as you can tell by the name, a certain number of bases is being inserted into the sequence. And this is so-called a frameshift mutation because everything else that follows afterwards will be different. Because whether we have 1 or 2 bases inserted, every single of the following amino acids will be different. And those mutations can be quite drastic to whatever organism of interest we're studying. And then also deletion, where we have a similar thing, where 1 or a few bases or larger deletions happen, and all the amino acids that follow this mutation will be different, which can affect the properties of a protein quite drastically. So to give you a bit of more context and examples as to when such events can happen is, for instance, a famous example of substitution is sickle cell anemia, where a single base substitution leads to a change in one of the amino acids of hemoglobin, which leads to this hemoglobin not being able to form the structures that it should, which then decreases its ability to bind oxygen as well, and also changes the shape of red blood cells, which is why we call it sickle cell. Then we also have avian sarcoma leukosis virus, which is a very interesting virus which infects primarily birds and chickens specifically. And what this virus does is it inserts a piece of its own DNA into the DNA of the host, which is a chicken, for example, which leads to frameshift mutations that make the host develop symptoms in response to this virus. And finally, we also have cystic fibrosis, where a deletion of a base leads to this genetic condition which causes dysfunctional lungs in the patients. So overall, the point I'm trying to make here is that within those protein coding sequences, although most of the time humans do acquire mutations that are silent, if there is a change in the amino acid sequence, it can lead to very drastic consequences for the person who's being affected or the organism that's being affected. And it's very important to understand exactly what kind of mutations you'd like to study, or in what way you want to change the gene in order to be able to achieve the results that you'd like through gene editing. So what is the definition of gene editing precisely? And according to Wikipedia, it's an alteration of the genetic material of a living organism by inserting, replacing, or deleting a DNA sequence, typically with the aim of improving some characteristic of a crop or farm animal or correcting a genetic disorder. And this definition is quite valid, but as we will hopefully discuss at the end of this talk, sometimes the aims of gene editing can actually be more harmful than beneficial, and it's definitely something that we want to consider about this technology. And what can we use gene editing for? And the applications are extremely vast, as some of you guys might probably know. It's a technology that has really reshaped the whole field of biomedical research in the last 10 years. But the major 3 sort of fields in which gene editing is being used a lot. First is basic research, where we alternate DNA, for example, in certain cells or mice, in order to identify the function of a certain genomic region. Then we can also have therapeutic genome editing, where we have in vivo genome editing, where we deliver those tools directly to the body, or we can have ex vivo genome editing, where we edit cells outside of the body and later deliver them to patients. And the latter, actually, ex vivo genome editing, has had a huge progress lately, where some of you might know that a new gene editing based drug was approved, I believe, last year by a company called Vertex Pharmaceuticals, where they develop a therapy against sickle cell anemia and beta thalassemia, where they take stem blood cells from the patients, then edit them, and then give them back to the patient to improve production of healthy red blood cells. And finally, there's also bioproduction, where we can engineer cells to produce useful compounds. So, for instance, in the current, I would say, climate, it's extremely applicable towards creating antibody therapeutics, which are becoming more and more popular with immunotherapy and other things. And this has also seen a lot of progress lately. So, how do we scientists do this genome editing in the lab? And although there have been various ways of doing this at the beginning of the 21st century, now the main technology that's being used pretty much by every single friend of mine who's doing wet lab research is CRISPR-Cas9. And this technology won the Nobel Prize in Chemistry in 2020. And it was developed by two brilliant scientists called Emmanuel Charpentier and Jennifer Doudna. And I do have to preface by saying that although they were the people who kind of discovered the property of this technology to be able to act as very accurate molecular scissors, the actual CRISPR complex was discovered a few decades prior by scientists who saw that genomes of some bacteria have very peculiar repetitive sequences which are resembling of viral DNA. And what they found is that actually bacteria use this sort of system for recognizing a virus when it injects its own DNA into the cell and then to attack it and chop it up, if that makes sense. And then once there's a new virus that comes in, the bacterial immune system again chops up that DNA and inserts it into its own genome so that next time the same virus can attack it, it will be able to recognize it. So originally this was found as so-called immune defense system of bacteria. But the two scientists, Emmanuel and Jennifer, they were able to actually modify in a way that enables us to precisely and accurately edit genes. And so the basic setup of this technology is a protein called Cas9 and a guide RNA. And Cas9 is a protein called nuclease that is able to cut DNA in very specific positions, while guide RNA, as the name says, it basically guides this protein towards a certain sequence and enables it to cut it. And once there is a double-stranded break, we can have two outcomes, which is either we disrupt the sequence or we can also insert it. And this depends on sort of which exact goal you're using this technology for and what's the aim of your experiment. But I also have to say that although this is sort of the classic model of CRISPR-Cas9 system, there have been many new other systems that have been developed where, for instance, there's only a single-stranded break or where the system is specifically targeted to a sequence to change just a single base by deamination, or where the Cas9 nuclease domain is actually inactivated and this protein is fused to a GFP protein. So that enables scientists to also visualize very specific sequences in DNA, for example, during replication or their spread across chromosomes and other things. So although this diagram shows kind of the very basic original system of CRISPR-Cas9, the system has seen a lot of change and evolution over the last 10 years or so. So if that's something that you're interested in, I definitely recommend looking it up. And so now I also wanted to show you guys a video that I think explains quite well how the technology works, and it kind of gives a moving image of what actually happens in the cell.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAN. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Yeah, so I just want to give you an example of a very sort of simple and fictional experiment that a scientist could potentially do in the lab. And before we do any experiment, of course, we need to first pick a very precise research question that we want to answer using the experiment that we're planning on doing. Then we want to pick a suitable model organism. In terms of what is being used widely, at least from my knowledge, is you can either study something in a cell line, you can also study something in an animal, for example, mice, or you can also, the most recent sort of model for studying biological process is organoids. And so depending on what kind of question you want to answer, you need to pick a suitable model organism for studying it. And that also requires a lot of sort of ethical considerations as to, for example, whether you really have to use mice in order to study something. Then we want to pick a suitable gene editing approach, whether we want to introduce a point mutation or we want to knock out the gene, so completely switch it off. And we also want to pick a suitable control that would help us compare the results of our gene editing to establish what goes wrong in that particular gene, for instance. And as I said, I wanted to give you a very fictional sort of setup where, for instance, if you're a clinical geneticist and you have a patient who's come to see a doctor with symptoms resembling sickle cell anemia, and genetic testing has identified a mutation ACT to ATT, which changes threonine to isoleucine amino acid in the hemoglobin gene HBB, specifically within codon 13. And as a scientist, I want to investigate how this mutation affects the shape of cells. So here we have a very precise question, is how does this mutation affect the shape of red blood cells and their function? In terms of our model system, we would want to use, obviously, cells. More specifically, would want to use blood stem cells because we can't edit red blood cells directly because they don't have a nucleus, as some of you might know. So we want to start from the precursor cell, which is a stem cell. And in our case, the gene editing tool would be used in order to introduce the single point mutation and compare how healthy cells would be different compared to a cell with this mutation. And so the first step in this experiment, assuming that you have a lot of resources to already have the cells in your lab, to already have all the kits in your lab, what you want to do is to find a suitable guide RNA for the sequence that we want to edit. And for this, what I wanted to show you is how to use a website called Benchling. So this is what Benchling looks like as you create an account and log in. And the website itself also has quite a few helpful tutorials that you guys can follow if you want to check it out for yourself. But I'll just here talk about the specific scenario that we're trying to look at. And to do that, I want to just enter the folder and then create a new page for CRISPR, and more specifically CRISPR guides. And then the first thing that we need to enter is the gene that we'd like to target. So in our case, it was a hemoglobin gene. So I just enter HBB. And genome is the human genome, which is what we want, Homo sapiens. Gene HBB, this is the region on the chromosome where this gene is residing. And this is the most standard transcript. We don't need anything fancy. So then we'll click next, and it loads it onto our system. And again, we just want guides for base editing. So all the classic sort of setup, nothing fancy, classic PAM sequence. Then we'll click finish. So now what we see is a sequence of HBB gene of the human genome. And as we can see, it's a very, very long sequence that spans a few thousand bases. But as you know, a lot of the gene sequence has many introns, exons, UTRs, which are not being transcribed into mRNA and not being translated into an amino acid, into a protein. But what we're particularly interested in is, as we discussed, codon 13. And so, which resides here, we have this threonine amino acid. And what we want is to basically generate a guide RNA that would target this sequence specifically. So what we can do is pick this region, let's say, of the first exon. And this is the target region from 2375 to 2466. And then we can click on this menu here design and analyze guides, then finish. And then we can click create, which will basically give us a bunch of various guide RNAs that could target various places within our selected region. And so from what we can see here is, if I show it like this, a bunch of various guide RNAs. So for example, this guide RNA will target this region, this, etc., etc. But the guide RNA that we want specifically is the one that would change this codon 13 from T to I, which is changing, which would have been ACT to ATT, which is exactly what we want. But something else that's very helpful about this software is that it gives us a base editing score and an off-target score. So for base editing score, we kind of want something that's as high as possible. And in our case, if I go back to this is it. Then the base editing score, as you can see, there are two numbers. One is 0.5, which is for this amino acid base highlighted here, and 13.5, which is for this highlighted here. So compared to the desired target, which is this codon, there is a small, tiny chance that there is also a mutation here, which is something that would have to check for potentially while doing our experiment. And then we also have our off-target score, and if I remember correctly, the higher it is, the better. So usually scientists aim to have an off-target score of around 40 and above. And we have 36.9, which is okay, which seems like the sequence would be reliable enough to target this specific sequence that we want. And so, yeah, so this here that we have above our DNA sequence is the sequence of guide RNA that we can use for our gene editing target, which we can then export. And then basically the way it's normally done, you can order it from a company that would send this guide RNA specifically to you, and then you'll be able to perform your experiment and change this base directly. So now we have a suitable guide RNA for the sequence that we want to edit. We send it off to a company and they ship it back to us with a CRISPR-Cas9 system, which we can use. Then what we do next is we deliver this CRISPR-Cas9 system into the model organism, or the model system, in which case is blood stem cells. And there are many ways that you can insert this system into the cells, but one of the popular ways of doing is, for example, viral transduction, where you have a loop of DNA encoding Cas9 nuclease and this CRISPR, the guide RNA sequence, which are then inserted into the cells, which then translate our Cas9 into a protein and make our guide RNA for us so that everything is now in the cell. And then we grow stem cells in a specific medium, then forces those stem cells to differentiate into red blood cells. And then we can do different experiments to address our main research question, which is, for instance, what is the shape of those cells when the mutation is present, or how well do they bind oxygen, etc., etc. So now that we can harvest those mutated cells, we can see how they affect shape and properties. And as you remember, I mentioned that we should always, in science, have a suitable control. And in this case, from talking to my friends who work with blood cancer specifically, a simple way of getting healthy red blood cells could be just asking for a donor to donate some blood, and you can just use their red blood cells, for instance, to also look under the microscope and see what the properties are like. And so, yeah, as I said, this experiment is very fictional and oversimplified. Actually, those are my friends who do use gene editing tools on a daily basis know that it's always tricky because you can have off-target effects, in a sense that you can still have the mutation that you want, but you can also have mutations in other places which are not ideal, which could mean that, for instance, your guide RNA is not specific enough. So you should work on creating a more specific sequence to make sure that nowhere else in DNA is being affected by it. Then also growing stem cells alone is quite a vigorous process, making sure that they grow in a suitable medium so that they differentiate into red blood cells rather than, let's say, white blood cells is also difficult. So yeah, I hope this just gave you an idea of how in science generally we try to answer certain questions, specifically using gene editing. And aside from the uses in the lab in research, gene editing has plenty of other uses. As I mentioned, we have now a few targeted medicines on the market that use gene therapy to fix certain mutations and help those affected by certain genetic disorder. Um, then a lot of scientists are also working towards creating sustainable biofuels or genetically modified microorganisms that are able to, for example, degrade waste much quicker or in a more sustainable way. For instance, instead of producing CO2, producing useful byproducts that can be used for something else. Then we also have disease-resistant crops that are being modified, for instance, to become more resistant to be attacked by certain species that are detrimental for farming. But as far as I know, in that area, the restrictions, especially in the EU, are very stringent. So there are not that many GMOs that are being sold in the supermarkets. Then also new industrial products, for instance, bacteria that can produce useful proteins at a very large scale, such as antibodies or, let's say, insulin, things like that. Then the challenges that come with this technology are also just as great as their advantages, because the first one that is the main reason why we still don't see CRISPR-Cas9 being used routinely in humans is safety, because the risk of having an off-target mutation is still relatively high, or the risk of, for instance, passing down that undesirable mutation to the offspring is also undesirable when we don't know its effects. Then we also have ethical concerns regarding who would be able to benefit from this technology. For instance, if, well, as we all know, in the US, for instance, any sort of therapeutics are very expensive. And so if you wanted to have access to a super new novel gene therapy, you would also have to pay a lot of money, which is not everyone can afford. So there's also, the issue of trying to make this technology as accessible as possible. And then we also have challenges in regulation and policy making. And there is an international agreement between scientists that CRISPR-Cas9 must not be used in human embryos. But as some of you might remember, maybe at least that was very big news for me when I was doing my A-levels, is when in 2018, a scientist from China created the first ever CRISPR-Cas9 babies, which, if I remember correctly, were twins who were genetically modified to be resistant to HIV because one of their parents was an HIV carrier. And that created a huge scandal in media, in the scientific community. I highly encourage you to read up on it because I found out recently that that same scientist is now actually back in the lab. So although there are certain policies and regulations in place, it's very important that they're actually being listened to and scientists follow them, because then it can lead to some tragic consequences. Although I think the babies are still kept a secret. So we don't really know what's actually happened to those children who were born from this technology. But yeah, I highly recommend you check that story out if you're interested. And finally, yeah, I hope that you guys found this talk helpful. And I tried not to make it too overwhelming or too technical, just to give a general idea as to how people like me who are doing a PhD in medical sciences use gene editing to answer certain scientific questions. I have left the link for BenchLink, which is the website that I used to create the guide RNA. If you'd like to check it out for yourself, you can come up with guide RNAs for any sequence in the human genome that you want. I also wanted to share a review, which was published in Nature, which is called the CRISPR Toolkit for Genome Editing and Beyond, which explores the topic a bit more in depth, and also an article on ethical issues associated with gene editing, which I think is something that's very important to consider about this technology. And finally, Jennifer Doudna, one of the co-winners of the Nobel Prize, recently published a book on CRISPR-Cas9 and gene editing in general called A Crack in Creation. I personally haven't read it, but I've heard quite good things about it. So if you wanted more of a lighthearted sort of popular science book to read in your free time to explore this concept in a bit more depth, then please do check it out. And I'm happy to answer any questions that you might have, whether it's about gene editing or studying biochemistry at university or doing a PhD. Yeah, I'm happy to answer.